continue from phosphofructokinase 1. Let me write it. Phosphofructokinase 1. Okay, so this is our topic, right? We usually call this big name as PFK1. Okay, so that it's easy to just say it, right? Now this is very important enzyme. Now you must ask me, like, why isn't hexokinase the rate limiting enzyme? This hexokinase, right? Because glucose 6 phosphate has many possible fate. That's the thing, right? This enzyme right there, glucose 6 phosphate, once you add the phosphate group on the glucose, this can actually enter into many different cycles, right? This is like a gateway. This can go in many directions. Yeah, it can go into, first of all, glycolysis itself, right? Glycolysis. It can go either into glycogen synthesis as well, right? Glycogen synthesis, right? It can also go into pentose phosphate pathway. So pentose phosphate, right? So it can go into these three pathways, right? And the thing is that how will it decide? That's the turning point here. That's the question here. How will it know that I should go to this pathway, or I should go to this pathway? How will it commit to going to glycolysis or maybe synthesis of glycogen? How will it know? That's the thing what our hero, Mr. Phosphofructokinase, does actually, right? The PFK1, this enzyme, this exact enzyme, actually changes the story. Now, what does it do? So we have fructose 6 phosphate, right? This glucose 6 phosphate or fructose 6 phosphate, whatever we have. What happens is that after talking to glucose 6 phosphate, right, it adds this biphosphate, one more phosphate, okay? That's the thing. After this step, the glucose is more committed to glycolysis. So PFK1 actually is just an enzyme that makes sure that your glucose 6 phosphate is not running away to another story. That's the thing. So PFK1 adds what? Adds another phosphate so that it becomes biphosphate. That's it. Okay. So PFK1, once it interacts with glucose or fructose, it actually adds another phosphate. And then what do we have? We have fructose or glucose. I hope I'm not wrong. Or glucose. One means at first position of the carbon, 6 means sixth position of the carbon, and bi means 2, biphosphate. So that's the end product we have now. What we have now is a biphosphate thing that is super duper committed to glycolysis. Okay. Now we have this, right? This is just commitment. PFK1 is just a committing enzyme. Make sure that your glucose, this glucose 6 phosphate here, right there, actually goes to the glycolysis itself. It doesn't just wander off into glycogen synthesis or pentose phosphate pathway, right? Now, must be asking me, well, how does it happen? Well, it interacts. What does it do on interaction? On interaction, it actually puts one more ATP, okay? So because commitment requires investment, right? The cell asks, do I really want to metabolize this glucose? If yes, then the cell invests one more time ATP, and then the cell, then the pathway proceeds itself. Now you can ask me, why PFK1 is the rate limiting enzyme, right? Why hexokinase is not, and why PFK1 is the rate limiting enzyme? That's a very cool thing to ask, right? Now let me write you. I'm going to tell you something mind-blowing. What I'm telling you is that it's a rate limiting enzyme, right? First of all, it's a statement. Now you must ask me, what is rate limiting enzyme? First of all, does it mean that it can slow the it can slow the reaction itself, or what is it? Well, rate limiting enzyme, it does not mean that it can slow the reaction. It means that this PFK1 enzyme actually determines the overall speed of the whole pathway. Okay, that's the meaning of rate limiting enzyme. Rate limiting enzyme means it determines the overall speed, right? So it determines, it's the decider, it's the main guy who decides the overall speed of the whole reaction. Okay, now what about regulation of PFK? Who takes care of PFK? Who tells PFK stop, right? Now regulation, like PFK answers to only one question, right? PFK is like, PFK, is anyone superior than PFK? Like, is there any serious, like, superior guy? Does the cell need ATP or not? Well, everything, all the possible regulation of PFK enzyme actually depends upon the amount of ATP if the cell has. If the cell has enough ATP, then PFK will work, because PFK is also just investing the ATP, right? PFK is putting the phosphate, right? Now, if you don't even have the ATP, then how the hell the PFK is gonna even work, right? So the question is that, does the cell have ATP? Does the cell have ATP? That's the question that decides PFK. That's the question. That is the question that decides the fate of PFK itself. Okay. If yes, I have ATP. Yes, I have. Then of course, glycolysis will increase. Speed is gonna go up. Yes. So speed goes up of glycolysis. Yeah, I can invest energy, right? I have ATP. No, I don't have any ATP. Then what's gonna happen? Then we're gonna have the glycolysis is gonna be stopped, like not stopped, but slow down, right? Slow down, because we don't have enough this ATP to invest, right? So you must be asking, tell me more about it. Because I don't want to tell you more about this right now, because this will come later on, right? Right now I'm just introducing you to these characters, right? Cool characters. So that was the first irreversible step, this glucose, this thing right there, right? Let me just outline this thing for you. So that's an irreversible step number 1, right? This phosphofructokinase is step number 3. Okay, right now we are talking about irreversible steps. Okay, let me just step 3, right? Usually the whole glycolysis has 10 steps. Okay, you must think, wow, this video is long, because I want to talk everything in details. Like, think about them, like, deeply. Why? You should be able to answer all the basic questions about them, not just memorize it. Yeah, the goal of science is, I think, to answer the most fundamental questions about, why, like, why is this even happening, right? Why it's going in this direction and not that direction. We should be able to answer all of the questions. That's why it takes a lot of time and a lot of energy to think about them, right? So now we understand step 3, step 1, right? Now I'm gonna take you to the step 10 journey, right? This is the last step. This is also irreversible. All of these step 1, step 1, step 3, and step 10 are irreversible in the process. Okay, so step 10, now the final irreversible reaction, right? This step is called pyruvate kinase step. Okay, pyruvate. You must thinking, I know this word. I think I heard about it somewhere. Well, pyruvate kinase efficiency hemolysis. Why? Because pyruvate is the final step. If pyruvate kinase step fails, you essentially do not have glycolysis. If you do not have glycolysis, your RBC is gonna burst up. They're gonna die. This, why they gonna bust up, Shashwat? Because this Na potassium ATP is gonna fail, and then we're gonna have hemolysis, right? So yeah, so pyruvate kinase, PY, pyruvate kinase, right? That's the final irreversible step, right? So what happens is that we have this reaction, right? What's the reaction? We have, okay, well, what's the initial reaction? We have PEP, right, turns to pyruvate, okay, pyruvate, right, and we get ATP as well in the final step. That's important, right? Now I want you to notice something. Now at the beginning ATP was spent there, right? Now we are finally getting the ATP back, right? The question here is, why is it even irreversible? Because PEP contains a huge amount of Gibbs energy, right? PEP, this little thing PEP contains enormous potential energy, right? Converting it to the pyruvate releases so much energy that the ATP can be synthesized, right? So this PEP, breaking this PEP, changing that PEP to pyruvate, releases a lot of large negative Gibbs energy, right? So this PEP is huge in energy, and that's why it's irreversible. The question of reversibility and irreversibility depends upon the amount of Gibbs energy a reaction has. Why a reaction is reversible? Because the energy, energy that is involved in the reaction should be like close to zero. The Gibbs energy should be zero, but the reaction should be very stable. If the reaction is not very stable and we get a lot of energy, or we put a lot of energy, then it becomes very tough for us to change that reaction again and again. So that's the key idea of reversibility, right? So again, we come to this question, that energy drink question, right? Why energy drink increases pyruvate kinase, right? Why does this energy drink increases pyruvate kinase, right? Well, energy drink actually, right? First of all, what's energy drink? Energy drink is just glucose, sucrose, and caffeine, okay? Right? So which irreversible enzyme increases activity? The question will ask you this. The answer is pyruvate kinase, right? You don't want to memorize that, because once you drink this high glucose intake, what's going to happen? Let me just make step by step, right? How about that? Even better. So what's going to happen? You take a lot of, insane amount of glucose, right? This glucose is going to do what? This glucose is going to increase your what? Insulin, of course. How? Well, by beta cells, right? I hope you know that. And even if you don't, it's okay. It's nothing like, it's not rocket science. So insulin increases, right, after glucose. Now you can ask me, why does it happen? Because insulin depends upon glucose, right? So more glucose enters more insulin sensitive tissue, so more glycolysis, so more glycolysis, right? And if more glycolysis, then greater flux, right, reaches to the final irreversible step, that is PEP converts to pyruvate kinase, or I should say pyruvate kinase, okay? So that's the holy grail right there. So last question that I want to ask myself is that, when do these enzymes break exactly, right? When do these irreversible steps break? When does it happen? So when can these things stop? Step 10, right? It's a good question to ask, right? So whenever we want to study about something, we want to ask ourselves, right? Hey, why? Okay, you want to understand why a car works. You should ask, your professor or your teacher that, when would this car fail? That'll teach you a lot about the car as well, right? So the thing here that we want to think is that, first of all, let's go to the hexokinase, right? Now, question here is that, why would hexokinase fail? Just imagine why. It can fail, right? Why would it fail? Because glucose cannot be trapped, right? So glycolysis stops immediately, right? So there are two things. First question is, why would hexokinase fail? Hexokinase would fail if we don't get enough glucose. First question, we have a faulty transporter, right? Glucose cannot enter inside the cell. That's point one. Question number two is, what would happen if hexokinase itself failed? Well, if it fails, then the glycolysis immediately stop, right? Because that's the step one, right? So that's the question for you, right? That's the answer for you. Another question is that we can think, when would PFK fail? Well, PFK is going to fail if we do not have enough ATP, if our cell is depleted of ATP itself, right? So yeah, the PFK would fail because PFK depends upon investing the ATP itself, right? So PFK fails on that situation. And you can ask me or yourself that, what would happen if PFK fails? If PFK fails, then the whole equation is gonna slow down or even stop itself because it's a rate limiting enzyme, right? It determines the overall speed of the whole reaction, right? So that's important, right? Because PFK fails, committed step blocked, right? Most glycolytic flux stops, glucose may be diverted to maybe synthesis pathway or pentose pathway, right? Maybe it will go to pentose phosphate, maybe it goes to glycogen synthesis, right? We need PFK here to say, hey, glucose 6-phosphate, go to the glycolysis, okay? What about pyruvate kinase? When do you think pyruvate kinase would fail? And what would happen if pyruvate kinase fails, right? If the pyruvate kinase pathway fails, the ATP, right, this thing will drop, right? RBC loses the ATP, the sodium potassium pumps fail, the membrane will become like super duper rigid, not flexible, right? And then what are we gonna have? Here we're gonna have hemolytic anemia. Simple stuff, right? So step number 1, step number 3, and step number 10 are all irreversible steps. What are these? Hexokinase, glucokinase, PFK, and pyruvate kinase, okay? Hexokinase and glucokinase just basically ask, what they answer, right? They answer the question that, should we capture the glucose or not? That's the thing they ask. PFK ask, should we commit to glycolysis? And the last thing is pyruvate kinase. What the hell is pyruvate kinase doing? It actually just harvests the ATP and finishes the glycolysis. So each irreversible step represents a decision in this metabolic story, right? And it's not just a chemical, just blah, 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 right? So that's the thing. So that's the end of part 3 actually. And I couldn't record it because I don't know, something went wrong in my recording. So that's part 4 gonna be. So in next part 5, we'll go through every remaining glycolytic step. That is step 2, step 4, step 5, step 6, step 7, step 8, step 9, right? We'll explain everything, okay? We'll understand all the story, right? We'll understand how NADH is generated, why ATP appears twice, why every downstream reaction occurs twice after the split, right? I hope you remember, this is a split. We have a 6-glucose, splits into 3C, that's a pyruvate. We know that pyruvate, we get 4 ATP, 2 NADH, right? So I'll answer everything, and we'll actually like see it happening in front of our eyes. I mean, most of the time I can see it happening in front of my eyes, and that's how I like it, right? So these steps are irreversible, the yellow steps. Now, okay, so I'm gonna upload this video. Take care, bye-bye.